I'm going to continue, and I'm actually doing this whole thing backwards because I'm going to end up with work that we did in my lab, 20 or 30 years ago. And it's still, to my knowledge, the one time that someone has done detailed quantitative comparison between a model and an experiment. Not just saying, here's a gradient, I've measured it accurately, and it must mean this, and I have my downstream genes, but actually, how does it emerge from noise? And this is one of my proudest efforts. I've yet to find anybody refer to it, though, in the literature, other than myself, of course. But that's what I'm say, that's where I'm headed. So I started with really simple, but I think complex problems where I talked about bursting and mRNA synthesis in E. coli. And I emphasized the importance for a biologist or an experimentalist to pick an organism that wasn't only tractable, but one where you could actually hope to finally nail something down in some kind of definitive way. And of course, you'll know from that presentation that it's still not really definitive because now we don't know quite why it behaves that way, but now we actually know how it behaves. And then I talked about the search problem and the crowded medium in the cytoplasm and the progress we've made there. And I think that there's a very interesting question for those people that are interested in transport and motion in the cytoplasm. And there's an interesting way to look at it. And I talked to one of you about this over tea, I think. So, and the question is, if you see molecules moving through the cell, and they seem to have directed motion to them, it looks as though they're going somewhere. And the obvious thing is if it's kinesin or dynein, then you can poison the cell and you can prove this. But what if it just seems to be moving and using some kind of bias thing, and then all of a sudden this behavior changes, and then it either speeds up or slows down, but it continues on its way. The tendency there is to invoke a magic force of some sort and start looking for a molecule that does this. But in the field of diffusion in matrices, you know, there's a huge number of people out there that are experts on watching things go through mazes. This is what groundwater people do in geology. And it's well known there's a threshold. So if you make a maze with a certain fractal dimension to it and you put a particle of a certain size in it and watch it go, it'll behave, you know, just the way you expect. It's subdiffusive. You can do all the parameters. But then if you change the actual fractal nature of it, just change the dimension a bit, there's a surprising transition at some point. So there's this great, all of a sudden, it's behaving differently. And if you're looking from the outside and you don't know this, you start to invoke all kinds of magic forces. So if you have transport things going on in your lab or in your head, there's this literature out there, and it comes from another discipline, and this one happens to be in geoscience, where percolation lattices have been explored both experimentally and physically for a long time. Easy experiments, after all. You just need sand in a box, and you can pour colored water in and watch it go. So that whole question of, and maybe this is what Rob was saying yesterday about seeing is not believing, because in this case you start to draw the wrong inference about quite a bit. And so I talked about those problems, and I went on to the search problem and talked to you about some work that Tom Coleman in my lab has just finished, and he's moved on this year, showing, I think, in a preliminary way, but I think it's really solid, that when a transcription factor is made at a site, it's more concentrated at that site for some measurable time period as it diffuses away to find a target. And we modeled that, and I showed the model briefly. Rob pointed out that I had two K1s in it. They had different functions, and I said, well, they're different K1s. So that's a biologist's way of getting out of these things. So I'm going to talk today now about work that's been going on and some thinking in my lab for quite a while. And that's a question of slime mold biology. And I'm going to show some movies because I really continue to believe that seeing is believing. And I'm showing them because I want to emphasize some of the really complicated problems that are still completely unsolved and maybe unsolvable. That's another question. When you have a reductionist agenda or a modeling agenda, how deep can you go and how satisfying can it be? And my view of theory, just to chime in with the others who are theorists and I'm not, I like to think of theory as a way to guide my experiments, to make some predictions, knowing, of course, that most of the time it's going to collapse. in biology at least. And biologists in general don't like that. There's a famous modeler, Hans Meinhardt in Germany, and he spent a lot of time writing quite beautiful papers on stripe formation in Drosophila and all kinds of things, polarity in neutrophils, did all of this work. He's published a couple of books on it. And at least in his own crowd in Tübingen, his reputation's not very high because, in fact, when you get to the details of how stripes form, they don't seem to fit his models. And so that somehow annoys people, whereas it doesn't annoy me because I see it as somehow deepening your understanding even when it's wrong. So I also look to models to deepen my understanding. I'll give you another anecdote. Since Rob was going on about this, I think I've earned the right because I listened to it, and gainfully. So I've bored my own faculty over the years with our pursuit of Turing equations. How many of you know about them? How many of you know about his famous paper? Yeah, well, art, of course. He and I must teach it at least once a year. So I bore them about it because they say, but wait, these have been around a long time and we still don't know if there's molecules. We still don't even know if it is molecules. Maybe it's long-range inhibition through neural circuits, or maybe it's a buckling phenomena. And I would say, and I always say, well, but wait, isn't it magic to you that these general equations also sort of describe the universe? It's not just biology, but they also, you can use them to do this and that, and it looks like the Fokker-Planck equation, so on and so forth. Well, there is a kind of experimentalist mind, I think, that doesn't want to know that because it kind of interferes with the experiments somehow. Well, anyway, I guess these things are all matters of taste. And I have good colleagues. I don't want to knock them. I do want to knock them on Turing, though. Anyway, so I'm going to do some review for you because I'll say again that when I talked to the physics graduate students and postdocs, I only found one that had a biology course. And they're all doing physical biology, the ones that I hang out with. And they learn the snippets they need to know. But I don't know what snippets you know, so I have to just give you a very brief outline. So I'm going to be talking about Dictyostelium. It's an amoeba, and I'll show you this amazing movie that Rob Cooper made in my lab about a year ago. But I just want to remind you that when you get to the eukaryotic cell, they're highly decorated. This is just a multi-stained thing. It's from this great book, a great compendium of stuff. And you see right away that it's got a cytoskeleton. And that cytoskeleton is quite elaborate. There's bundles of actin filaments. There's green fibers, there's yellow fibers, there's a nucleus, and these are all dynamic. So this is a fixed specimen. You're looking at it under a fluorescent microscope. This nucleus is already 4 or 5 times the size of an E. coli. So it's pretty big as well. But it's not just a bag of cytoplasm. And we know quite a lot, in principle at least, enough to draw a cartoon in this textbook about how cells crawl. That's what I'll be talking about. But just to give you a very, very brief overview, they stick to a substratum. They send out something called a lamellipodium. And they have a cortex made of actin. And they figured out how to trundle along by making and breaking contacts here and by trundling actin back to the tip and recycling it to the back. And each step, the protrusion comes, they adhere. There's a contraction at the base where myosin II is, and I'll show you some pictures of Dicty too, movies of them where you can see the myosin II and you can see the actin. And this is the back, and there's a contraction and keeps pushing it forward. Now, of course, that's a cartoon, and it's not an area that I work in or even know a great deal about, but I do know that there's a lot of it we don't understand yet. So it's an active area of cell and molecular biology. And here's a fish keratocyte, and I think it was taken in Julie Theriot's lab, I think, a co-author of with Rob in the book that you all know about. And if you just take a scale from a fish and put it down on a cover slip under a microscope with a little bit of saline, this amazing cell will crawl out most of the time and head off. And again, this is the lamellipodium. It's huge. It's just full of actin, and the back here is full of myosin II. And to me, the most outstanding thing about this is the rate at which it moves, but also it goes in a straight line for very long distances. So if you're if you like to think of models to explain things like this, right away you think, well, here's one edge and here's the other, and somehow it's doing a waddle like this, and there must be some feedback here so that when this one speeds ahead, this one will catch up. So it probably waddles like that a bit at some molecular level, but we don't know. But there they go. And here's a famous movie that you can find on YouTube. It was taken many years ago by David Rogers, and he simply took a drop of human blood, his own, put it under a microscope, and then put a little bit of Staph aureus bacterial cells. Gives you a good feeling for the scale too, I think. So that's actually 2 of them. And watch this guy go. And this movie actually raises another issue. It raises lots of issues, actually. You see the big lamellipodi in front, and it can smell these guys. And we even know what it's smelling. Bacteria release things, waste products, and different species that go after them, amoeba and so on, sense the waste products. It will catch it. There, it's caught it. And it keeps going.
Why is there no more recent, better version?
I don't know. Probably I haven't looked.
No, I have.
Have you? Okay.
I actually went and pursued, where that movie came from and stuff. And
Couldn't find it.
find it all.
It's really
It's so
interesting.
fantastic.
Yeah.
You'd think somebody would have tried to, make it.
So there is, so from a physical point of view, there's another really interesting question here that occurred to me. I'm sorry, I'm going to just give you anecdotes because whatever the frame shift is coming from Princeton started to catch up with me. So you'll have to cut me some slack. But I had shown this movie to some high school students. And the next day, I was out sitting in a hot tub that we have in our backyard, contemplating the bush in front of me. And there was a piece of debris fell in the water. And it came towards me, so I reached out to grab it. So you know what happened, right? So what's the Reynolds numbers going on here? So what happens, of course, is you do this, and you send a jet of water out, and the leaf just gets further away, and you can spend the rest of your life chasing the darn thing around. So you have to have another strategy. And when I did a little bit of looking into the literature, and Rob might know more about this, since we've got real biologists here and cell biologists as well, I don't know how cells do this at this low Reynolds number. How do they ever grab anything? So that got me started on the project I'm going to talk to you about today. And so this part has 4 parts. I better get going. So this is the thing. There's a big literature in Dictyostelium about how they sense gradients of cyclic AMP, which is their chemoattractant. And there's a prominent model out there that's widely believed. There's also a very nice experiment done by Van Oudenaarde, which makes me, at least, doubt the model completely. So you notice in the neutrophil case, you didn't see a round cell that then broke a symmetry and headed towards a bacterium from a signal, right? What you saw was the cell already had a nose. And it sniffed something here, so it turned around and headed like this. So that I see many cell types that I know a little bit about as nothing fancy. No signaling to give you capping, and there's a stochastic event here, and you get a little budding, and it grows through positive feedback. I see the cell, the neutrophil, certainly, I think, and I'm increasingly Dictyostelium, as the kind of model I tell my students a primitive man would come up with, or a primitive woman. Which, what would you do? What would they do? They would say, well, there must be a little wooden arrow, a little stick in there with a sharp end on it, and it just uses that to tell its head from its tail. When it smells something, it just swings around. So it's real simple. It's built in. It doesn't reconstruct it afresh every time it has to move. So I started to study this because a really talented graduate student from physics came to me and said, what should I do if I want to do biology? And I said, well, there's all this Levy-Walk literature. Do you all know about it? Most of the mathematical part's not hokum, but the biology under it is mostly hokum. I could tell anecdotes about that too. And I have no friends in the field, so I don't have anyone to insult. So I said, well, why don't we actually ask what single dictyamoebae do in the absence of signals altogether, right? So this is Liang Li's work, and this is on her thesis. in an attempt to describe it to the layman, she said, well, Dicty is like this person, and they're playing hide and seek in a big field, and there's clumps of trees. There's another person here. You may not be able to see it from where you are. And the question is, what is the optimum strategy to find this person? Okay? Well, it's going to depend on spacing. It's going to depend on numbers of trees, all kinds of parameters. It will depend on time. For instance, you can convince yourself fairly quickly that if you have an infinite amount of time, the best thing to do is to go in a perfectly straight line and then turn your sensing width and come back again. And just keep doing that because you'll cover everything fairly efficiently. But it'll take you forever. Another way to do it, and the way we do it, that's the way I used to play hide and go seek, I'd run here and look around, and then dart over here and look around. That's the obvious way one might do it. Another search that's efficient but takes forever is a pure Brownian search. So you just go out and you do standard old Newtonian, Brownian, Einsteinian walks, and sooner or later you'll uncover it. So she's showing a little simulation here. This is the same number of step lengths. And so this is the area you cover just with a random walk, mean x squared versus 2 dt. Dark blue naturally because there's a lot of resampling, which you don't want to do in a hide and seek game. Here's Olivier Benichou's model off of the same problem, which in his PRL paper is really neat because he likens this to a dog looking for food, which is a dog sniffs. And so here's the sniffing thing, and then runs a ways and then sniffs again. And then basically when you model this, you choose the wait times from some distribution. And you can get quite rich behavior by doing that. So the actual behavior here doesn't have to be Brownian either. It can be something else. So this has become, like many things in science, an entire field of theorists, basically. And there's a Levy walk, which was much in the literature. And the dangerous thing about this is papers in cell biology started to come out and claim it's a Levy walk, it's a Levy walk, it's a Levy walk. But when you look at the data set, it's the weakest possible support for a Levy walk. Yeah, I'm going to digress a little bit more here on the dangers of believing something as sweet as this. So a Levy distribution looks just like the diffusion equation, except you raise the power of time to some other exponent, and then that gives the distribution a very long tail. In fact, it has no second moments, so that you can have infinitely long excursions here. And there's a theoretical literature that shows rigorously in one dimension that is the most efficient way. But that's it. So why has this lost favor with the editors of Nature? Well, it's because the principal data came from albatrosses in the Galapagos. And this clever biologist had a little sensor on their feet that sensed salt water. And then he had telemetry and a satellite to keep track of where they were. And you know, albatrosses will go out and coast forever and then land for a fish, so he could get that signal. And the data, you could construct a walk like this. So this was a big deal. And then someone pointed this out to the biologist, who was basically reading much different kinds of papers, and he said, oh, they sit in the nest for long periods of time too. They never leave land, they just sit there. So that's what this is. They weren't moving at all, they were just sitting there in their nest. And then I was invited to one of these conferences, and put your hands up if you're getting totally bored by this. I can ignore you. So I was at a conference in Copenhagen, and there's another one, howler monkeys. Well, they certainly follow the Levy walk. This is all big papers on this. And the biologist who gathered the data showed the data. It was really beautiful because sure enough, there's this Levy walk, long excursions, and then he showed the map of the area under the data, and there's a lake here. And he said, and so, you see, monkeys don't like to swim. Well, here's Dictyostelium. This is hard data and a soft model, and that's what I'm going to go through today. So this is of one of the tracks. Well, actually, I'm sorry, it's a simulation based on the same number of steps and the same step size. So this is a Dictyostelium amoeba now that has no food anywhere, and it's a 10-centimeter dish. So, you know, it's thousands of cell diameters away from anything. As far as we know then, there the spacing is like, we put them down and we keep looking till we find one and then we follow it. So they do this walk. And I'll give you the answer, but then I'll show you the data. Basically, they do it by waddling, as Henrik Flichberg calls it. They go a little bit like this, and they persist in a little bit of a straight line, and then they do this. But then on top of that, there's a drift term, and on the long scale, it looks like Brownian motion. Right? So then they'll do this, and so it turns out that this walk is about 2 times better than a straight line walk. I'm sorry, than a Brownian walk. It's not perfect. It's certainly not what you can do in 1 dimension here, but it's real data about a real animal. And then we're modeling it. And so then I'm going to go on and talk about the model a bit. It's really simple. And we've done some simulations, and Rob Cooper has published a paper or 2. Then I'm going to talk about the properties of many of these cells when they have to signal to each other to form a new organism for the next generation. And then I'm going to talk about recent advances that come from work done by Thomas Greger and Satoshi Sawai, which is a nice lineage because I'll be talking about Satoshi's work with me in my lab and then the work he did with Thomas in his lab. So it's a generational thing going here. All right, just movies again. So they're pretty efficient at finding stuff. There's actually the corpse of an old amoeba here. So they also have this kind of touching behavior, you'll see. It's sort of like a little either cannibalism or a little ceremony over the, who knows. So you can imagine there's a lot to explain here. And when they go for the bacterium, they're sensing basically, well, they could be sensing quite a few things. Dying bacteria secrete pteridines. They secrete little peptides. And here's another one that Simon Flichberg made. This is a tiny E. coli colony. There, divided. So I'm going to let this run to the end because there's another surprising phenomenon that no one had suspected. See, they're nibbling at it. These other ones come in from the side because there's a gradient of junk that they've sensed. So that's easy enough. But now look what happens when they finish their meal. There's sort of one thick-witted one that stays there, hoping for the best, and the others have all gone off in another direction with some kind of walk and some kind of worry about where the next meal's coming from. Then I'm going to go on, and the next step will be, I'll have time for this, don't worry. So Rob just put this Petri dish under a dissecting scope with a camera. And so there's a layer of E. coli here. It's about quite thick. It's pretty rich medium, maybe a 0.5 millimeter thick. And this is covered with Dictyostelium cells at different stages of development. And there's a wave here where they're all humped up going in this direction to eat more E. coli. And there's several things I want you to keep your eye open for. You will see clumps of cells that are signaling, and you can see waves traveling out from their center. You can then see in some other parts of the image cells crawling in towards the center, and they're surfing up those waves. And then you'll see these things we call slugs that are moving around apparently rapidly and apparently with some purpose, but they have no neurons, they have no brain. There's only 2 cell types. There's the stalk cells, and then there's the other cells that will become spores. And then you'll see them finding a little dry spot, and they know it's dry and the light's on, so then they ascend and form this little fruiting body with a ball of spores at the end. And my agenda for many years has been to try and discover interesting features about as much of this as I can. So these are called social amoebae for reasons that are pretty clear, I think, and you find them out in the woods. And there, you see the waves pulsing along here, and you'll see some more down there. I had all this set up with an iPad because with Keynote on an iPad, you can touch the screen and you've got a little pointer to move around. It's really cool, except that I couldn't get it to work, so I'm back to my old, okay, we see that? So that's essentially, there's a lot to explain there. And just to give you another sense of scale, this is taken a picture made by Chris Wagner, so there's just a piece of bark on a tree, and there's the mature fruiting bodies. And there's all kinds of theories about why they do this, but essentially, no one knows. So if you are interested in collective behavior, this is a good one. Yeah?
These waves we're looking at density waves itself. Is that why we're able to see it in this movie?
No, actually what you're seeing is they're a density wave of a sort. The amoebae are contracting, they're doing this. So when they're signaling, they have one refractive index, and when they're moving, they have another. But the thing looks just like a smooth sheet. You're not seeing humps, for example. Okay. So this is what Liang did. She put the cells down, as I said, and then followed them. And the nice thing about this is you can get beautiful data sets. So I think one of the problems with areas of molecular biology that I'm not attracted to is it's hard to get good data sets. You end up with, a few samples. And that's a great thing about bacteria, of course, is you quickly get large data sets. I mean, a physicist, of course, wouldn't work on a system where they couldn't. You can already see the waddle in this one. That's why I chose it. It's got quite a persistent direction here for this shorter time. And it managed to get there by turning left and right with a bias, of course. Another great virtue here is that these guys can do this for very long periods of time. So here's 10 hours. You can follow them. They're starving, but they have enough stuff stored away that even in the absence of bacteria, they can just keep going. So we typically set these up under a microscope and follow them for 10 or 12 or even 20 hours. And this is important because we don't want to be looking at a change in state of the amoeba. We want to just watch its behavior. And this is just a track of the velocity, or speed, really. Especially my physics graduates, they never shut up about the difference between velocity and speed. And I always say, I know that. But I still, in weak moments, I know one's a vector and one's, yeah. Anyway, you can see, and you can also see if you just look at the differential here, that there's little fluctuations about mean behavior. So this is a good experimental system. There's 3 tracks shown there. And then here's how we analyze it. The way any of you would do it, you simply have to have some MATLAB-based way of automating this so you can go through many tracks and get lots of data. And essentially, this is it. We have a tracking program, so we call this a turn. We call this another turn. And obviously, we call this a left turn and this one a right turn. And we keep track of these. And so Liang has these huge data sets. There's always a question, if anyone that's looked at cell movement literature, there's always a question of when does a cell turn? Because as you know, as the time gets shorter and shorter, you get to a point, Zeno paradox-like, where who knows what it's doing? You can't tell turn. So you have to make some decisions about whether this is really a turn or not, whether this one is. And we do that in our case by just changing the parameters on the algorithm and seeing if it makes any difference to the bottom line in the model. Okay, so here's the data, one of the pieces of data. And these are left-left turns. These are right-right turns, 2 successive ones. Here's the right-left, and here's the left-right. And you can see just from looking at the quality and the amount of the data, there's a clear distinction between successive lefts or rights and the zigzag. And it's almost exactly 2. And we've done this in other amoeba from other species. I'll be talking later about Polysphondylium, which is evolved away from Dictyostelium probably something like 500 million years ago. And it does the same thing. It's better at it, so that the scatter plot looks even better somehow. But this is the quality of the data. So I think this is just the right thing for a physics student to get started on. She's now got her PhD, so it was a good thing to end on, too. All right, so now I talked about the long-range behavior. So we now have a persistent walk, but it's a persistent random walk nonetheless. So we waddle, we waddle, we waddle. We go off the screen and we come back and we keep. But you notice now that there's a drift in the behavior over the long term. So it doesn't have a memory that extends very far back. It extends back, actually the left-right memory, we can show from analyzing the data, we can stimulate it very nicely with a simple coin flipping algorithm. So it only remembers the last turn. We know it doesn't remember back the second and the third last. But nonetheless, just by drawing at random, you can get walks that persist this length of time. And then finally, the data on a bigger scale turns out to look like Brownian motion. Okay, so here's some more of the data. Like many of my papers, the referees didn't like this. And I'll tell you why. Because we have all our data here. And they said, well, if you're right, what is this one doing? And I said, well, we don't know. But that's what it did. And so I got into this argument with a referee over this, and I said, you know, if I show you a straight line, can you tell me it's not diffusion? And he said, oh, I see your point. So that did the day. That did it. So here's just a mean squared displacement versus time. And here's something with a slope of 2, which would be ballistic. And here's the behavior. Here's our data. And here's Brownian motion here with a slope of 1. And here it's plotted in a standard way, I think, in much of the literature. So we just divide by time here. So it's the mean squared displacement divided by the time versus the time. And there's a characteristic crossover. And so here's an average line in blue. And so they persist for about 30 minutes, and they go 5 or 6 body lengths in a straight line before they turn. And so this is just a summary of what I said. And we have another paper in physical biology that goes into this in great depth, because Henrik Flijberg is, I consider to be the world's expert on diffusion of all kinds, and a very sharp analytical mind. Yeah, I'm going to shut up now for a bit too.
That crossover time gives the time scale over which that memory persists, is it?
Yes, up to right to the crossover time. Yep.
Okay. Typically, how much is the number of runs it takes in one particular direction?
Could you use the microphone? There's a fan going on here, and it's a little hard for me to hear with the fan.
Typically, how many number of runs it takes before it's crossing that turnover time?
So that has to be answered I think in the following way, because the run lengths are not all the same, right?
Right.
And so there's different variable run lengths, and then and variable turns. And so roughly I've forgotten the mean number, but it's something like 5. Okay. And the cell's there's this length scale on there somewhere on one of those slides.
All right.
So obviously the cell has organized its cytoskeleton the way that cartoon, somehow it's like that. And then the question is, how do we explain this? Do you have questions about this? So I think it's pretty simple, but if it isn't, please ask a question. Yeah. No, we do the centroid of the perimeter. Most of the literature does the centroid of the mass, and with anything that has pseudopods going out and around, that's a very bad idea when you think about it a bit. It can be quite deceptive. And we're after pseudopods anyway, so the perimeter makes more sense. Yeah. I mean, if we changed anything in this field, it was people now measure perimeter and not the center of mass. Okay, so let me go on to how we're going to model it. And we have an effective model. And it's something that would occur to almost anybody, I think, but there are some subtleties to it, and it's not all that easy to pick the right parameters and think that you're onto something. Now, this is a model in the sense that it's, I hope you'll agree with me, it's not just a summary of the data, because I don't think it's that. It actually makes predictions, and the literature was full of raw data on Dicty moving around. So people knew where the pseudopods were going, how many, when one appeared here, where did the next one appear? None of it was explained. So what Rob Cooper was able to do with the model was to go to the literature and plausibly explain the literature. So I thought that was a bit of one of the functions that you can use a model for in biology. So it goes back many years to a suggestion by Hans Meinhardt, and actually originally by Alan Turing, and I'll get back to Turing later. Basically, this model has a memory built into it. It has a local inhibitor. That's why the bar is like this. So it's not a short-term activation model. It's a memory model. So this is like the end of the pointed stick that I was talking about. So there's a memory in the cortex here, and it has a certain lifetime. It can form and decay. There's a local inhibitor here that's governed by the same system that controls this. And the obvious reason for that is to stop. It diffuses away. Doesn't have to be diffusion. It could be a cortical change that propagates like a diffusion term. Okay, that would be possible. See, prevents new pseudopods from springing up nearby, because that's not how the cell behaves. It tends to send out, if you notice the movie, it's doing this. It's not bifurcating, and so on and so forth. And then there's a global inhibitor, and the reason for that is that to get the wigwag, the waddling motion, basically I'll give you the answer to this. The waddling motion is pseudopods here. We're going to call this the grandfather or the grandmother pseudopod. And another one will form here. It tends not to form there again because there's the memory still there, although the memory's decaying. So it'll form nearby but not back at the same site. Okay? So that's how it works. And occasionally, the memory molecule fires, or whatever it is, fires somewhere else, maybe here, with some probability, and that gives us the longer-term drift. So once a pseudopod forms at this end, and this one decays away, then the cell heads this way. So it'll go this way. These have lifetimes. The global inhibitor is there all the time, and it acts as a threshold to stop firing of memory molecule, and particularly the short-range inhibitor. And then they start decaying. And now it decays first here, nearby. We get a new memory position. So we might form a pseudopod here. So then we head this way. If we exceed the threshold of the global inhibitor, say over here, this is a stochastic process, then we form a pseudopod, and gives you then that longer-term drift. And we also use this to explain chemotaxis. So I'm not going to show you so here's the data that Rob used. And you can actually see, I think this is actually, Rob's a very modest fellow, and I think this is much better than he thinks it is. So myosin is in the rear of this crawling cell. And you can actually see from looking at it that it's waddling along. And actin's in the front. You can see where the pseudopods are coming out. And then Rob has a really nice way of analyzing the data. And this is in a methods paper, but it's already used quite widely. So another problem with tracking cells from a practical applied science point of view is that, do you call this a pseudopod? Do you call this a pseudopod? There's all this question of categorizing events. And Rob does this by a hierarchical clustering method, and that runs independently of me or Rob. You put the data under there, and you use a hierarchical clustering algorithm to come up and then just label them, rather than say, I see one now. And so here's a composite of a track. Really lovely stuff. I can say that because Rob's not in the audience. So here's the model. I've already explained it to you once. But as the walrus said to his crew, what I tell you 3 times is true. So there it is. There's the global inhibitor, the local, and there's a memory. And then along comes the parent. We're going to talk about grandparent, parent, and child. And you can see what's happening. So the memory now has decayed. Oops, I keep pointing on the screen. That's not good. The memory now has decayed away here. This one's dominant, and so the cell is heading this way. We can now form a child over here because the inhibitor has gone down to the point where it can fire again. And meanwhile, we're preventing firing around the rest of the perimeter, roughly speaking. You know that's true only to a certain point because you know that the zigzag choice is only 2-fold above randomness. So this is set up for that, basically. And here's a simulation on the left, and there's some experimental data here. It comes from the literature. It's not a great fit, but it is what it is. It comes from papers published by Peter van Hastert, and there's Rob's comparison there with his model. And here's another one. So now we're comparing parent to grandparent from the literature. And I think it's a kind of pretty good fit. I think it's fairly satisfying. And I didn't bring the equations with me, but they're pretty standard, involving diffusion of the short-range inhibitor, and then a delta function for firing, and then a threshold function for the global inhibitor that keeps things suppressed most of the time, right? So that's basically it. Now I want to turn to another, yeah, sure.
Wouldn't it be ideal if that angle was actually 90 degrees, but here you're seeing a distribution that sort of, where the peak is between 60 and 90?
Do you mean in terms of modeling or in terms of strategy for the organism?
In terms of strategy for the organism.
Yeah, I hadn't thought about that. Would it be better? Would it be more efficient? So you do this, and then you do this. I know how to settle it without just answering. I think it might be, but I'm not sure. So here's how we think about it. You think of the amoeba as tracing a path of some width, and when it turns, it resamples in this angle here. And so the function that says this is twice as good as Brownian motion is simply looking at resampling.
Oh.
And so if your idea might, no, you do more resampling that way because so if you just turn a little bit, you just resample this little vertex
smaller
here.
area,
You do this, that included angle, I think it's got more material, so it's less efficient. I think, but I'd have to draw it on the board. Yeah, in the back.
So are these components, actuator and inhibitor and memory, are they components identified, or it is?
No. No, that's one thing. Rob wanted to start down that route, but you know there's, this Rob pointed out how many receptors there are in E. coli. Well, Dicty has maybe 100 times as many. And there's protein kinases all over the place, and there's lipid synthesis molecules. And we have good genetics, so we could start sampling those. But imagine how, well, I can tell you it's hard to systematically screen through all of the candidates. And I don't believe, myself, I don't think right now that's a fruitful thing to do. But if someone has a high throughput, massively parallel super machine, then that would be the way to go. But no, we have nothing. I will show you, though, where we do have a lot of information. That's coming up in a bit, a different related subject. Okay, so here's how you transition to chemotaxis. There's a nice feature of the model, and we actually have some simulations to show how effective it is, and we can compare how effective it is to known data in the literature. So the chemotactics index is just a measure of how straight you go towards a source. So if you're going away from it, it's either 0 or -2 or something like that. And as you deviate from the straight line towards the source, the value changes and it drops from 1 down through here. So essentially, when there's no gradient, we have this. These are a bunch of separate tracks now. They're different simulations. The colors are different tracks. Basically, it looks like Brownian motion, or I'm sorry, it looks like the persistent motion and the waggle walk. 3% gradient, we begin to see motion towards the source, 10%, 50%. And here's a lot of data, and it looks pretty much like the literature data. So I want to talk a little bit about then, in this case, you'll notice that this is done as a kind of a symmetry breaking event, right? You've got something like that, and you excite locally, and you get a little patch of memory here, and then away you go. And that was because this is the easy way to do the simulation. So the real question, though, is, does Dicty actually do this, or does it always have a pseudopod waiting somewhere and then just turn? And surprisingly, the literature on this, I don't think is very good. And the literature, I think, has been misled by the nose for the following reason. If you do an experiment like this, you don't do it with a Dictyostelium cell, because it's so pseudopods everywhere. What you do is destroy the actin cytoplasm with a drug, latrunculin, and it forms a nice spherical cell. So, aha, you're physicists, this is perfect, a spherical cell. What happens? Well, we now can label up actin or we can label up myosin, something like that, and we can put a pipette tip out here and diffuse a chemoattractant, and we watch a patch of our labeled stuff form here. So this looks pretty good because it looks as though there's a real symmetry breaking event, and then the pseudopod will form here because the actin's in the pseudopod. Now there's a problem with this experiment. Some years ago, the Van Oudenaarde lab, when he was still at MIT, actually repeated this work, found a patch, right? And then moved the pipette tip around, let it decay, moved it around, and stimulated it here. Where do you think the patch formed? Well, you know, right? It formed over here. And it turns out that this is the distribution of where it forms as a function of where you put the pipette tip makes it look very much as though the whole orientation is already internal. And this has led to a big controversy in the literature because in neutrophils, it's been known for a long time. Neutrophils, you can see them heading this way and they sense a bacterium and they just, like the taxi driver last night, this terrifying ride back, he just kept turning in the middle of the highway like that. Yes?
If you started the pipette somewhere else, would you see the memory there to begin with?
Well, of course, when it's spherically symmetric like this, the first spritz that you do, you don't have any landmarks, but it's true the first spritz does organize something. But then you let that evidence decay. So what I actually think, with all due respect for my colleagues, I think there's an awful lot of data selection going on because these experiments are hard to do. Very hard to do. In fairness to the literature and my colleagues, I think that's a possibility. Because as you point out, what about spritzing here? Shouldn't you sometimes find a patch over there? And I don't think you do that. It's just that once it's here, it's really nailed in as a memory. And then it doesn't matter where you spritz after that. And that seems, you can't tell whether it decays away forever because these cells take a long time to recover from latrunculin. So Rob Cooper started out to do an obvious thing, which was to repeat the Van Oudenaarde work, remove the latrunculin and see which way the pseudopod formed. But we could never do that. So I don't think we understand this very well yet. All right, second part. So this is, to get to your question from the back, this is a way now of really nailing down the molecules and learning something quantitative about these waves as they propagate. So let me show you another movie. This is taken from a project that Satoshi did in my lab. We built a robot, and in this robot we could look at up to 600 little petri dishes, each one with a layer of slime mold cells, and then we could categorize their behavior, and then we could look at mutants, because now we have this array, and we could guess at what the players would be, because that's a good literature on that, coming from many organisms. So there we go. Now this is a movie I show my introductory biology students, and I say to them, as I said to you the other day about another movie, if you don't find this exciting, you should find another career for yourself. It shouldn't be biology anymore, maybe doctoring or lawyering or something like that. So this is really rich image, right? Whoops. So those territories are big. There's about 100,000 to 1 million cells in each territory, so they're signaling over long distances to each other. And you notice that there's spiral waves doing this, and there's circles. Not so many here, but I'll show you more. Look like targets. They expand. They start in the middle like a heartbeat. They do this. Now these waves, actually when they collide, extinguish each other. Another feature of this movie. A good question. So basically this is just transmitted light movie, but it's dark field optics, and we do frame subtraction. So we only see things that change from frame to frame. And you're actually looking at contraction of the cells as this wave sweeps by. The wave is now known unambiguously to be a cyclic AMP wave. And I'll get to how we know that towards the end. So
In the movie, there's some structure.
exactly. Exactly. So what Rob's pointing to, and you've noticed, is at the center of these spirals, there's a pinwheel. The cells now start to crawl in. And they're going to form a fruiting body here, a la the last movie. There, look at that beauty. Yeah. There's also a change, the apparent color changes, right? So things that were dark turn white, or we don't know why that's, like a lot of things. We don't. So how does this work? So if you imagine an oscillator producing a circular wave, which is how our heart beats, they expand out and disappear. And they go boom, boom, and another one. In our heart, it's a calcium and electrical wave that goes around. So it goes boom, 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 like this. So when you have a circular wave, you need an oscillator at the center to drive it. And so it does this sinusoidal thing. But with a spiral, you don't. This has, of course, profound consequences for humans because a very common type of heart attack is so-called reentrant waves. And the reentrant waves have this spiral quality. And once you've got them, they turn faster than the target, so they take over from the target patterns, and they no longer need an oscillator. So just take a look at this. Gray is excitable, so this is just the layer of cells again. It's just a little cellular automaton model. So there's a layer of cells here. Gray are the excitable ones. That means they can respond to the cyclic AMP that's being secreted. I don't think I mentioned cyclic A yet. Different slime molds use different factors, as a matter of fact. The white guys are refractory. They can't smell it anymore. And the black are the excited ones. Okay? So when I run this movie, you'll see that in the front of the nose here, there's always fresh cells that are becoming sensitive to the signal. So it just keeps going around like this. And then out here, it does this. So understanding this sort of thing is really important for just to understand development in this organism. And you see how it goes. But I've put some other examples down here. When you fertilize a xenopus egg, you also want to coordinate everything around the cortex, and you do that with a calcium wave as well. If you put a little IP3, which is down the pathway further, you put it in, you get spirals building up all over the surface. And when you have a heart attack with these reentrant waves, the whole surface of the wave starts to quiver with these spiral waves going everywhere. And when you defibrillate somebody, I know this is a digression, but it's the same equations, basically. When you defibrillate somebody, you're shutting down all those spiral waves to the rest state, and then, like a miracle, the oscillator's still there. It's true in most people anyway. It's true in Dictyostelium, too. We've done this experiment. You shut down the wave. When things come back, the oscillator is still where it used to be, and it can get going again. We have a little perfume bottle with cyclic A in it, and like that. I have a slide of that. Actually, it was done with Ray Goldstein and Hyung Lee years ago. Okay, so I could philosophize about this endlessly too. So the question is, how much do you have to know about the details to be effective physicist in biology? I don't know. Don't learn too much is my view, because it'll inhibit you a lot. So there's two views of how this oscillatory circuit is set up and how the waves propagate. And I'll tell you the old view that comes from work in my lab and also from theoretical work, beautiful work by Albert Goldbetter in Belgium. It is simply this. There's a receptor for cyclic A, so it bounds cyclic A here. Then cyclic A does all of this in the cell. It's needed for correct message readout. It somehow regulates the ACA, adenyl cyclase gene. The adenyl cyclase gene secretes transmembrane protein, secretes cyclic A, and this cycle goes on. Now, you have to restore it to the ground state, too. Remember, there's cells there that are refractive, there's cells that are moving, and cells secreting. So you have to now degrade the cyclic A for a period externally, and that's the role of this phosphodiesterase. degrades to cyclic A. And then you have to do something about this, so the cells also secrete an inhibitor of this. So this cycle forms an oscillator. And that's the center for the oscillating wave. So that was the standard view. But there's also a cycle down here, consisting of, this is a phosphodiesterase here. This is a protein kinase, ERK2. There's another protein kinase A here. There's an activator of it, and there's cyclic AMP involved here. And so the view arose and appeared as a paper in Science that said this is the oscillator. This is responsible for propagation. And we know that because when we knock these out, we can't make waves anymore. Okay, so that was the assertion. These mutants in these genes were picked up as flat layers of cells that couldn't form those streams. So that was not unreasonable to believe that. And so the authors of that paper said, this really isn't the important part of the cycle. So we started to look at this. This is another question of seeing, not being, believing. But we can make real progress on subjects like this in these cells because we can do the quantitative analysis of wave frequency in time and space, and I'll show you how we did that. But then we can use mutants to dissect the models that we've come up with. And I think that's really important. And then we can model it and test it again by using mutational analysis. So I think that those go hand in hand, and don't worry about this function here. This is so you know how to do Fourier transforms and look for power spectra in any data set. Unfortunately there, when you do that, you lose one of the dimensions. But this is another way. It's called wavelet analysis. You might be familiar with it. It's a way to preserve more information, but it also makes modeling assumptions here. But I don't want you to look at that. I want to show you another movie. So here we are. Again, waves spring up. You can see target patterns there. And spirals spring up. If you watch this a couple of times, you'd notice the spirals take over the targets. But the important thing is you have a portrait now of this whole movie. And that was important for us because now we can make quantitative statements about differences between conditions and between mutants. And we can also look for patterns in these large data sets we got with our robot without just staring at movies and saying, oh, yes, that must mean this and that must mean that. So here's the wave frequency plotted here. There's time here in minutes. And so the cells aren't doing very much. They're sitting there not moving. In fact, they don't move until here. It's another virtue of this system. They're just lying there. And now they begin to go through whatever cycle they're going through, and the waves start, and they grow more and more intense, and they fall off. And there's another signal out here, and that signal is when they're crawling towards the middle, streaming to form this fruiting body. All right, some mutants. Are there any questions about this? Am I going too fast? Yeah. It's dense. It doesn't have to, I would like to be able to tell you precisely about occupancy of the space. They're almost touching each other, but they don't have to be. Well, there gets to be a point where you get outside the diffusion range of the cyclic A, and so things don't work very well. But you know, it works over in a pretty astonishing large area. Okay, so let's look at mutants. So I'm going to pick one here, ERK-B, which is in this other oscillator inside the cell. And there's some movies. And there's the wavelet portrait here. And you can see that one of the interesting things about the ERK2 mutant, although you don't see aggregates and fruiting bodies, you actually see oscillators. And the way you find them here is to take a little patch. It's something you can do now with good microscopy. You just take a square box and watch that. Whereas if you look at this, it looks like a bowl of oatmeal. It doesn't look like anything's going on. But look, the oscillation, even the frequency's not so bad. And here we just plot what's going on, right? But then another nice thing about it is, with the genetics, you can complement this. You can put the wild-type gene back and complement it, which is of the essence for a geneticist, and you restore this wild-type behavior. The frequency builds, decays, and then later everything starts to come together. Well, let's go on here. All right, so basically, it begins to suggest that although this does have something to do with the wave size and shape, it doesn't have anything to do with the oscillator itself that sets it up. So that's an important. Now I want to show you again, this is a quantitative analysis that Satoshi did. And so let me just show you. We have a way of finding out now how many of these centers there are in this field. And it's a fairly standard technique, I'm told. I can't myself do it. But we use a phase analysis to find a number of oscillating centers. So look what's happened in this mutant where you've decoupled cyclic A internally. You haven't destroyed the oscillators at all. The real problem is there's 5 times more than there should be. So it's very deceptive to look at that thing and say, aha, the whole thing depends on the ERK system. It doesn't. The lengthening of the wave and the long-range cooperation obviously does. But there's a normal one. The black ones are clockwise, and I've been searching for years for a mutant that only went one way, but don't think it's going to happen. So that's just a brief summary, and here's another brief summary. We've crossed out all those guys now by just using these mutants. And so the whole thing is driven by external cyclic A binding, and secretion. Now, related to this, are there any questions about this? Too fast? Hope it's not boring. I'm trying to make the main point, though, that there is a way of combining genetics, cell biology, and quantitative reasoning to deepen our understanding. Yeah. Yeah. More or less. It doesn't matter very much, actually. I'll show you that it was a very local thing. That, in fact, there was an expanding wave, and then by chance, a cell behind the wave fired prematurely, and so made a cut in the wave, and then that started to roll back on itself and form a spiral. So that was a very local model with a very specific gene in mind, and we had the mutant gene, so we published some papers. And Herbie Levine had a very different view of this, which I found really annoying. It turns out he's right. He had the right view. The right view is that there's sort of extensive asymmetries over here. They're not all starved to the same extent. And these asymmetries then give you a map. He called it genetic feedback. I never understood that because it's nothing to do with genetic feedback. But basically, because of the asynchronies, that this on a large scale gives rise to spiral waves rather than, I mean, it's not that difficult an idea probably, but this is a modification of his way of looking at it. So this is essentially the rate of cyclic AMP synthesis in space and time. So there's a diffusion term, it's degraded, and then it's released. And this is the excitability described in some way, that is going above this threshold and actually secreting cyclic AMP. And it has a kind of a prefactor here, and then it's dependent on concentration, and then there's quite a bit of fiddling here to make it work well. Well, we added one of these terms to explain the data, and it's kind of a nice model, and it is good at explaining the data. There's some simulations in the lab. It would take me a whole afternoon to derive all of this, so I'm not going to try. So by tuning this factor beta in the excitability term, we can account for quite a bit of the wavelength differences that we see in these experiments. And afterwards, I could talk about this to anyone, but I really want to move on a bit. So here's actually the simulations. So there's the number of phase singularities, those little spots that you saw, the gray and the white ones, as a function of beta values. And here's our mutants. This is wild type, 2 different wild type strains. There's the phase singularities per centimeter squared in this mutant. There's a lot, as you remember. Reg A has way too many. And here we complement. This is a regulatory subunit of another one. Basically, the idea is that the excitability is quite different in these strains, but the ability to form an oscillator is untouched. And by coupling the excitability levels to the oscillator, you can vary the frequency of the waves, how big they are, how fast they go. And, okay, your question. I have a slide. I knew you were going to ask this question. So here's synchronized cells. We can synchronize them, not very well, but we can do it. So here's asynchronous ones at the top. So this is a recapitulation of everything you've seen. So you'll notice that there's a couple of targets there, and spirals then arise from this non-uniform excitable landscape. And now we synchronize them and watch them in the cell cycle. So there's early G2 phase, so this is just before the cells are going to divide. So what we're trying to do here, of course, is make them all very uniform. So there's no way for the non-uniformities to feed back on everything else. So you see an interesting thing here. You guys would have guessed if I gave you an exam, like a general exam question, how do spirals arise? You would say because they collide, and that leaves free ends. And the free ends then, because of that other quality of always having their nose in fresh tissue to excite, that's it. But isn't it? You can see that from that picture. Targets collide, spirals never arise. And this is just another one to make the point, Anyway, I've done the conclusions for you. You obviously have understood it. Only one person's asleep. That's good. All right.
Can we look at the
Yep.
middle one more time?
Sure, you bet.
The video that you were saying where the targets collide.
Yes. The top one. I'm sorry. No, it's the early G2. Well, you can look at them both, Rob. Sure. Up here, sorry. Oh, no, those are target. I'm sorry, those are spirals. Here, yeah, they're already spirals. There you go. Isn't that interesting? So these systems are so, God, they've been evolving for so long. It's still an area where one can learn a lot, I think. And I think it has this more general interest, of course. So then the question is, everything we've measured so far is indirect. It's changes in refractive index of these layers. And nowadays you can use fret sensors to look at these guys. And so when Satoshi left my lab and went to Tokyo, he started to immediately tweak a cyclic AMP binding protein fret sensor. And it turned out to take him 2 years to do this because everyone says, oh, we'll just do a fret sensor and it'll bind this and we'll have a signal. The fact is you've got to match it to the KD of the system. You can't just put one in. And that took a lot of tweaking. But he did. So this is a movie that Thomas Gregor gave me. So these are just the cells signaling in a little drop now. And the question is, you saw those territories forming when the cells aggregated. The question is, are there special cells at the middle that do this? And the answer is no. There's a community of cells. And without the cells moving, that community drifts. It does this. Most unusual, you saw it, so it aggregates in a little drop. There's the fret sensor. And like many fret sensors, it kind of works the wrong way for an experimentalist. You start with a signal and you lose it as things happen. But basically you bind cyclic AMP, so you get a good YFP signal here, you bind it, and then you get low fret from the blue protein over here. So signaling drops. This is not really cyclic AMP oscillations. It's a phony thing done with injecting a dye. But here's now the relative intensity of a single cell pulsing, and it, like a miracle, has about the same frequency as the things I've been showing you. About 7 minutes, something like that. But let me show you this one. So now we're looking at internal cyclic AMP levels in this layer, this monolayer of cells. I just find this worthy of many looks. Now look, so they're aggregating, and now you could see that in the light movie too. So now so I'm working with a postdoc right now, and we have a fret sensor for adenyl cyclase. as well. So now we can do another step. Oh, there it goes, moves off. Okay, so what can you do with this? I already asserted what the problem was. How do you actually look at this? Do you see it as a true oscillator like the node in your heart? Or whether in fact it's a community property that somehow is like quorum sensing? And I'll show you that I think it's like the latter. And so we're going to do individual cells versus fixed spatial regions. So we're going to watch one of those droplets now. And we're going to follow individual cells and then watch what happens to them. So this is the data. You've seen some of it already from this paper of Thomas's up here. So early on, these are just many different cells. Those are the color traces. And with time, things ramp up, and you get this beautiful cyclic AMP signals. And now this plot is just a question of how long it takes for them to respond. So these guys were the first responders, and this level is the minutes after when you first saw some activity here. So this is here, and under everything I believed up to seeing this paper, I would have thought the center of the spiral would have formed here. Seems logical, but in fact, look at what happens with time. So it started over here, it's here now, it's back here, and now at this point, it's pretty solid here. So, how do you model this? Well, I don't think this has been done yet, but they refer to it as a kind of quorum sensing. About 10 cells involved in a small community, and it drifts around and competes with its neighbors. There'd be no cell movement here. That's the other thing that's really important. They're not going anywhere. They're just communicating with each other. Okay. 20 minutes, so now I get to what I consider to be one of the great scientific papers of all time. It's the last paper Turing wrote, and I thought I would put this in here for you because Rob mentioned things about modeling, and Boris is very interested. We had a long discussion over some wine last night about the function of models in science in general. And I love what Turing said. This is one of the most honestly written, forthright papers I've ever read. The second sentence is, this model will be a simplification and an idealization and consequently a falsification. But basically, hope it leads somewhere and clarifies the problem. And it sure did. So here's what Turing did. I've got time to sort of BS along here for a bit more. So basically, he realized that you could have two chemicals that reacted with each other in certain ways, and both of them diffused. And if one of them diffused faster than the other, you could set up spatial patterns. It's not that simple, but close. He did this with linear ordinary differential equations, and he solved it analytically, which he could only do on a ring. So there's no boundary conditions on the ring. And then I think, although nobody knows, I think he looked through a book to find an example of this. And the thing he found was Hydra. Because in Hydra, you can start here, you can cut this off, and you get a stump like this, and this stump will regenerate one of these. And the symmetry of the tentacles, that's little theta I put here, is quite extraordinary. I mean, I don't think there's a literature on mean and variance. That's something that would be interesting to know, but basically it's like this. And there's this simple model. It's the activator-inhibitor model, and I use A and H because this was developed independently and unknown to Geer and Meinhardt. They didn't know about the Turing paper. They did this in the early 70s, and it's an H for Heimstoff. It's an inhibitor. So the activator has this autocatalytic property, and it also stimulates synthesis of the inhibitor, which feeds back and shuts down the activator. And if you choose your kinetic parameters correctly and make the diffusion terms have the correct relationship, of course you want the activator to diffuse slowly and the inhibitor faster so that you get spacing between whatever's going to spring up, like here. And there's the diffusion equations. This is also, or was, it no longer is an industry. And so you put nonlinear terms in here to make it grow quickly and, to make it behave. So I think there's good evidence for this now in several organisms. And I want to spend this last moment telling you what we did many years ago, and then give you an idea that I've had in manuscript form for 11 years now, but I'm afraid to publish it. So I'm relying on Art and Rob to tell me after this whether I should forge ahead or just let it go for 12 years. So this is actually Hans Bode's work from Art's institution, and it's not quantitative at all. But what they did was make antibodies against the proteins that they could extract from here. They made a bunch of monoclones. And so the question is, what does the pre-pattern look like? That's what Turing was after. He said, look, you can do this, and you still haven't seen anything. No bulges, no tips, no arms, nothing. There's a pre-pattern under there. And he actually coined the word morphogen in that paper. Everyone should read it. It also is beautifully written. It says right at the beginning that if you're a mathematician, you should skip these numbered paragraphs. If you're a biologist, you shouldn't try reading these other paragraphs because it won't mean anything to you. And he also then shows that if you have the activator diffusing slowly, much slower than the inhibitor, you get standing patterns like my slime molds. But if you reverse it, you get traveling waves. And then he also shows that if you bring the diffusion coefficients closer and closer together, you didn't call it it's not really chaotic, but you get, the outcome is really uncertain. You can't tell what's going to happen. So he did all of that, just a big head. And he said he was embarrassed by the simplicity of the mathematics. So this is what Hans Bode showed. So this is a beautiful, this is an early stage. There's the final stage. These are single cells, so it gives you a sense of the scale of the organism, maybe a millimeter long, a couple of millimeters. And this is a transition you observe when it grows back. So basically, what isn't clear from this paper is clear, I think, in mine, and what I'm going to talk about now. You begin to form it, and it's all over this hemispheric tip, something, a morphogen. It's a protein, probably a glycoprotein. And then it breaks up, a bit motley, and then you see these tips, and then they just keep growing. This is the one we worked on. This is Kachin Greg's part of her thesis work. So this is another slime mold. I'll talk about it in a minute. It's polysphondylium. And when it's marching up the stalk to form a ball of spores at the top, it deposits behind nice little balls of spores. And these balls of spores form arms. So it looks like a little Christmas tree. And I thought, aha, this is just the thing I've been looking for. This is a question of choosing an organism to solve a problem, to look at Turing's stuff in more detail. So if we probe, we've done this now with probes for genes, we've done it with antibodies, we've done all kinds of different ways, at least two, I should say, not more than two. There's none of the expression of this stuff here, the blue protein. But then with time, as this begins to pinch off, it's everywhere on the surface. We've sectioned all of these. There's just no expression inside, so it's just on the surface. It gets restricted quickly to the equator. And the equator breaks up finally to form these radially symmetric patches. But it goes to an intermediate stage not shown here where the patches are there at a higher frequency and more irregular. What Kachin did was isolate genes. So these are all genes that will become stalk cells eventually, just by chance. She isolated genes that were known to be expressed in spores. They're everywhere in this, as this origin's growing. And then you clear this zone, and it's exactly symmetrical to this. So you lose the spore-like stuff around here, only on the surface, and then you begin to see the same thing. So I'm going to get to some quantitative data in a moment, but I want to say there's another organism where the actual players in patterning in one dimension are known now, and that's in Anabina, which is a cyanobacterium. When cyanobacterium grow, that's all the green stuff in the lakes that are not so aerobic anymore. As long as there's nitrogen source around, like ammonia, they just grow like bacteria and form long chains. But when they run out of ammonia, or an amino acid, say, they have to fix nitrogen from the air. And they can do that. But the trouble is, you know, fixing nitrogen is the Haber process. It's a huge free energy change, and it's not consistent with normal metabolism. So they have to form these little things called heterocysts. Doesn't come through very well here, but the arrows point to the heterocysts, and here they're stained with a GFP tag. And the miraculous thing is the heterocysts end up 7 or 8 cells apart. And it has this distribution. And now you can do good genetics with this organism. That's how you get GFP in. And now it's pretty clear that there is a lateral inhibition effect, and it's a small peptide, like a neuropeptide, that diffuses along and shuts down heterocysts everywhere but at the peak of this stuff. So and I want one more. So this is what's in this 11-year-old manuscript. You'll notice that in this case, and in the heterocyst case, and in Polysphendylium, these organisms do everything in one dimension. I mean, the current view in the slime molds was that there's a bunch of special cells in here, like positive charges, and they find their way out and they set up this pattern. But it isn't that at all. You have a universal pattern and then you restrict it. So this is true for many organisms as a general statement. The question is, why do you do this? So I'm going to tell you the heart of my 11-year-old paper. You do it because in one dimension you can faithfully use a Turing-like mechanism to get a reproducible pattern. When you go to 2 dimensions, remember now what happens in Turing is there's modes, like sine waves growing and competing. In 2 dimensions, finally, it'll settle down, but the nodes will be at different positions every time you do it. And you can show this easily by simulation. I've done many of them. And so I've given you one good example here of patterning in 1 versus 2 on the leg of a fly. Now, you can't see any of this, but flies have little hairs like we do. And here they are, and they're needed for something. I don't know what. And there's actually, if you do nearest neighbor analysis of these guys, you find out that there is some patterning there, but it's weak. It's a long way from a hexagonal or a square lattice. This was done in 1960 by a guy who worked at the Sheep Institute in Australia, actually, and he stained sheep embryos and did the same thing. This pattern, though, which is species-specific and has to be really regular, is laid down in 1 dimension in the disk. So you can guess what my paper's saying. My paper is purely phenomenological, but it says essentially that if you're going to do spatial patterning in an animal, and if evolution has to work, it's got to be reproducible. And that means you have to reduce the dimensionality as fast and soon as you can. So Drosophila's already done this. It's 1-dimensional. It forms bicoid here and to the back. And I don't want to get into this, but I believe that frog embryos and everybody else does this too. And worms don't gastrulate, so we don't have to worry about the worms. So let me quickly, now that I've given you this big story, so there's this beautiful organism, a really lovely Fred Spiegel picture. So you see the symmetries here, and the spacing is very regular. Here's a blow-up. These cells are still moving up. These guys are just, so this is what attracted me, right? You get the whole life cycle on 1 here, so you can do. We know something about the spacing. That was this cover in Nature. It's the only cover that's ever produced a slime mold picture for anybody. And what this paper showed was that to get this spacing, they don't count cells, because the diploid cells are twice as long as haploid cells, and one of these guys is a haploid and the other one's a diploid. So they're not measuring, one of the wits that refereed it said maybe they're counting chromosomes, but anyway. So here's how we did it. So here's how we looked at it from a Turing point of view. We simply had some various reagents, and we could stain for the pre-pattern. And in those days, we put it in a little capillary and rotated it like this under the microscope and took pictures and then sewed the pictures together with pinking shears. So this is a ball of cells at the base. You can see these antigens are around the surface. You can even see the pinking shear marks here. So this is around the surface of one that actually has some primitive little tips. And you can see the spacing's pretty good already. And then I was at that time fancying myself as a quantitative biologist. And I figured, well, I want to nail this down. So here's the actual trace. This is the Fourier transform of the data. And this is Fourier synthesis of the Fourier transform to show that we knew what we were doing. And the nice thing about the Fourier transform is you can use a Fisher's test, Fisher 1, not Fisher 2, to ask about significance of the frequency. And it's centered here. It's nonlinear, so there's not one frequency pops up, but the spacing is pretty good. And that's the little example that we used for that. Because you have to go fast to give the next speaker a chance. So here's a bunch all laid out. They're fixed specimens, so they're done in a slightly deceptive way. This is what we think time is, but we can't really tell because we fixed them. Only this one has visible tips. None of the others have visible tips. And here's the nuclei stained with a nuclear stain. So you can see that there's not much there. There's just background in some of these. And with time, you see patches coming up. So there's competition. Here's a model, Gero-Meinhardt kind of adaptation of Turing. Initially, you go super threshold, you fire, you get these peaks everywhere. There's competition between them, and we finally end up with 3. There's some panels taken from it. There's a Fourier spectrum of these guys. And then here's the same thing for our experimental stuff. So I still think that this is Jerry Burns' work from a long time ago. And I'm not aware of anybody else that's actually managed to say that, yes, it not only looks like a Turing mechanism, but it has the properties of one. So these are the people, various people. Kyung Lee is a professor in Seoul at the moment, Satoshi in Japan. George and Ling were senior technicians in my lab for many years. Pete Thomason's a molecular geneticist. He's at Cambridge now. Thomas is actually, when he was a graduate student at Princeton, we started talking about these things. So he's actually, now that I've collapsed my lab, I gave him my good microscopes, and I now have an office in physics. That was the bargain. Ned Wingreen, most of you know Ned. I have a few papers with Ned, including the ones on the waddling behavior. Liang Li is a graduate student in physics, now graduated. Simon Norielka is a real hardcore physical biologist, now in Zurich. Rob Cooper just graduated, and he's doing oscillators and synthetic biology at UCSD. George Guan Ling also worked here, and Simon's father, Henrik. It's very ingrown. That was this is the group of people that helped work on this project. So that's it. Thank you very much. Take any questions you might have. You're up next, Rob. Yeah. Could you wait for the mic?
Oh, yeah.
Or I guess I can move away from whatever's in there.
Yeah. So in the onset of collective behavior,
Yeah.
so the location where they finally form, all of them aggregate. So what sort of decides that?
We don't know. Because the latest data with the FRET reporter shows that what starts out as a cyclic AMP very active center doesn't persist. And now that center drifts, but the cells don't drift. It's just the activity's drifting. So they're inhibiting and activating each other in a way I don't think we understand. It's been modeled. That's from a science paper, some of it. And you can look it up. Just Google up Thomas Greger and take a look.
Yeah. Sure.
It might also be that the FRET reporter is not reporting maybe just fluctuations, and they're not really causal, and maybe when it finally settles down and begins to form a fruiting body, maybe that's when it's gone past a certain threshold. Well, undoubtedly that's true, actually. The main point is there's a lot of talking going on earlier in those patches, and they move around. Yeah, in the back.
So just out of curiosity, is it possible to engineer the cells in such a way that they have a straight path during chemotaxis?
They have a what part, sorry?
Straight path. They don't go zigzag. Instead, they have a straight path during chemotaxis.
Oh, they do once they start surfing on the cyclic AMP wave, then it's ballistic. They go straight up.
Okay. There's no clockwise and anti-clockwise motion?
Not on the individual cellular level.
Okay.
No, it's just a sheet, And the waves are, it's just like you sitting there and you see a wave coming towards you. And the question, one real question, of course, is how do you know you're heading in the right direction when you're sensing change in slope in a wave? And we know that many cells make a mistake. They'll go the wrong way. And they persist going the wrong way with small fraction. Of course, if you're a biologist, you think, oh, Darwin set it up that way because it has selective fitness to have stupid people wandering around somehow. You know, you can write a book on this stuff. Yes?
Yeah, I had a comment to make on the memory molecules that you mentioned during chemotaxis.
Yeah.
And you said you have no idea what they are.
Well,
But-
I do, but it's not a...
Okay.
I have too many ideas about what they might be is the problem.
Shouldn't they be secondary metabolites? I mean, like the quorum sensing molecules?
I don't think along those lines, but you want them to be secreting something, do you?
Yeah.
So one thing we know about this, so the paper where I showed the data on left-right choices, so there's many ways to model this. You can do like a self-avoiding polymer walk. You can do complete statistical treatments of various kinds. And the bottom line is there's no evidence for anything in the media. For instance, and in the experimental data, you might think, if your suggestion's correct, that they would find their old path and follow it, right? Or cross over, and you'd show some behavior as they crossed a trail or something like that. We don't see any evidence for that either, but yeah, I mean, who knows? Yeah.
So are two molecules are necessary for creating spatial pattern?
Yes, of the kind I'm talking about. And moreover, you can use more than two, or you can also think of inhibition as exhaustion of something, right? So you can get the longer range effect by exhausting the material you use to make the activator. It looks the same in the equations, but it's really just using one molecule. So yeah, I should have said that perhaps. So I want to say again, my paper that's 11 years in the making, I liken this, just so, I hope you'll go away and remember this, I liken it to a symphony orchestra. And I ask my colleagues in my department when I talk about this, what's the dimensionality of the instruments in a symphony orchestra? And well, some people get confused about this, let me tell you, because I say, well, there's only one instrument that's two-dimensional. They're all one-dimensional. Think of the violins, the oboes, the flutes, and everything. And then Mark Rose always puts his hand up and says, bells are three-dimensional. I said, no, Mark, they're not. They're two-dimensional. The timpani is two-dimensional, but how many people here know that you have to tune a timpani? In Berlioz, yeah, you might probably play timpanis, but in Berlioz, there's one of his famous thing where he has like 100 timpanis. They're all F at one point, and then there's something else somewhere else. And I'm told by timpanists that they're extremely hard instruments to play because if you don't tap it to the right place with the right weight, you just get a, you don't get the note at all. And most people listening don't hear a note anyway, they just hear a thump. So that's basically what happens. And there's no symphony instrument that's a big ball of lead on a chain that someone hits with a hammer. But of course, those are physical waves in hard media. Yeah.
Yeah, is it fair to understand that the circuit outside the cell is the one that is providing the oscillations?
Yeah.
And the circuit inside is actually the response to the amount of cyclic AMP in the environment of the cell, which is trying to kind of the logic circuit that is trying to understand as the response to the environment.
I think that's a good way to look at it. Yeah, you certainly need the internal one. I think the evidence for an oscillator inside is itself a little weak, but anyway, you do need those players to take that short range information, all those crowded oscillators, and turn it into longer range information. So that's pretty clear. All right. Next.